Martin and April took us through a very valuable clinical journey, and I will now try to take us through clinical development journey when it comes to chronic urticaria. And there are a few things to consider. First, we will look at the development space but from a different perspective. Then we would try to tackle some of the strategic considerations that are important for you, whether you're a sponsor who is developing or an investigator who is working on one of these molecules. We will obviously analyze protocols and discuss some of the operational challenges. So I want to thank my colleague. She worked on these 2 slides. These are intense slides, and the reason why I'm showing them to you is to start to appreciate the sheer volume of development we currently have in chronic urticaria space. The way we divided this is on the first slide, we're looking at biologics. As you can see, primarily those are monoclonal antibodies, and April already covered very well different mechanisms of action, so you can see that they're grouped there. And what is important to notice is that they're in different stages of development, some completed, some still going on. And when we continue on the second slide, we're now looking on all the small molecules that are being developed in the space again, a variety of targets and phases of development, as well as the status of the studies themselves. So what is this telling us? It's telling us that we are actually living in a very competitive space. So if you're developing in chronic urticaria, you have to keep that in mind. Patients are becoming not as easy to approach, right? Especially in the earlier stages of development, we want patients who are naive, right? So they haven't been exposed to any of this, so we need to keep that in mind as we're developing our studies. They need to be smartly developed, competitive, and provide you enough information to move forward, but not overburdened with too many endpoints that are going to make your trials so unattractive, especially in earlier stages of development, that they will go to a different trial. From a strategic considerations perspective, you know that our regulators have become more interested in us providing a significant amount of data to justify approach to indications such as chronic urticaria, especially in the context of how regulators perceive this disease. So it is good, and you'll notice that most of the companies in development actually have decent appreciation of what exactly is the target and how is it affecting the disease. There is also, in earlier stages of development, an important consideration is about biomarkers. You could leverage biomarkers in the earlier stages of development and allow yourself to combine what you see clinically, even if it's in a form of a trend, correlate with biomarkers. There are not that many good biomarkers, by the way, in the chronic urticaria space, but there are a few, right? Depending on what therapy you're developing, obviously IgE is going to be one. You can use D-dimer, you can use CCRP, so there are others that you can consider. The clinical outcomes are different. We are in a PRO world, right? So everything is reported by the patient, and that in itself carries some benefits and some risks, which we are going to discuss further. And then finally, because of how busy this space is we actually from very early on have to think about globalizing our studies. So we will be most likely in multiple regions, which is also important to take into account because the approach to investigation is different. Also, what I haven't seen enough is actually innovativeness in how we're designing studies. So are there opportunities for us to maybe approach earlier different types of urticaria by doing basket trials, right? There's also a question, is there always need to have a double-blind study in your early stages of development, or would it be enough to do an open-label small study to just see whether there's signal correlate with your biomarkers and then move on with a decent level of certainty? And then there is also an opportunity for adaptive designs. I would actually suggest you to read an article that was published by Innovaderm researchers on adaptive designs in rheumatology, because this is one of the indications where we can actually venture into this place, especially combining earlier stages of development. And then there are regulatory implications. For the first time, we're actually not with the derm division. Most of these molecules are being developed in collaboration with pulmonology, allergy, and critical care division. So we are getting to know other approaches, right? There are a couple of programs that are actually still with their division, and that's a matter of negotiation. If most of their program is within dermatologic indications, within FDA, there is that opportunity, but most of the companies are actually developing in a different, with a different division. There are also specific considerations related to working with other regulatory agencies. I will even point out to you that many companies in the later stages of development, they're differentiating the primary endpoints submitted to FDA and EMA, right? So it's different. So also something to keep in mind from a regulatory perspective. And Japan is becoming, as you know, increasingly interesting as a market overall, and chronic urticaria is no exception in terms of do we want to go there, not go there. So we have to take into account the PMDA's principles, which are sometimes quite different from what we're used to. From protocol considerations perspective, I mentioned to you that we live obviously in a PRO world, and these are most common primary endpoints. There is a small inclination towards using itch severity score weekly when submitting to FDA versus Urticaria activity score weekly for EMA. But you can see them but you can see UAS7 present on a lot of protocols submitted to FDA as well. And then we have a variety of secondary endpoints, right? Disease control related to UAS7, that is less than 6. This is defined as a well-controlled chronic spontaneous urticaria. Obviously, change from baseline in hives. This is a weekly score, similar for angioedema. Then you have obviously a high bar score, which is a complete absence of the disease. This is something that we're aspiring clinically to. And the one that is very interesting to me personally, as you've seen probably publications in that area, is how do we define minimal clinically relevant or important difference? And there are two criteria, right? So more than 10 points decrease in UAS7 or 5 points decrease in HSSSN. In addition to that, we have obviously certain endpoints that we're going to consider when we're developing in inducible urticaria space. So first of all, we're interested in proportion of patients who have a clinically negative provocation test. Then change in the provocation threshold is an important endpoint, and finally change in local itch, right, in the area of provocation. What has been variable, and now I'm speaking both from the literature and our own experience at Indero. I was just about to say Innovaderm, but I made it. Indero is that it's important to have a clear definition of what is threshold for eligibility at baseline. For example, for cold urticaria, it is different from sponsor to sponsor. Some sponsors put it at 10 degrees, some at 15 degrees, some at 18 degrees, so this is something that we notice is a variable. Also importance of how we're assessing itch, because this is localized itch, so it's very important that we guide properly the patient to get the reliable results. If you really want to be able to track the change in the threshold, you need to use STEM test. It cannot be tested by eyes. And enrollment targets are actually different for different types of chronic inducible urticaria. It's been also our own experience that cold urticaria is much more difficult to enroll than cholinergic or allergic urticaria. In terms of the inclusion criteria, this is important one, is what are we going to do with omalizumab, right? So are we going to wash out omalizumab, which is probably not recommended in earlier stages of development, or are we looking for naive, omalizumab naive, or are you interested in actually cracking that nut and trying to show that you can treat patients refractory to omalizumab? I think that can be a significant advantage. So you can approach it different ways. You can completely forbid it, you can cap it, terms of a clear definition of diagnosis, I would actually be very careful here because you're looking for patients who had continually at least 6 weeks of disease. It's not 6 weeks anyway, right? It has to be continually 6 weeks of disease with the occurrence of lesions multiple days within each week for consecutive 6 weeks in order to be eligible. And that's something that needs to be well explained in the protocol. And then finally, we are looking for having in the UAS7 more than 16 during this first 7 day prior to day 1. So that's the criteria to confirm that the patient is eligible to participate in the study. The other area of sort of discussion, I would say, is what are we going to do with our antihistamines, right? So what is considered a stable treatment, right? For how long should the patient be on a stable treatment for us to conclude that the patient is actually not reacting to therapy? Are we to do that before we confirm that it was sort of up dose, up to 4 times the regular dose for antihistamines? So all those are all questions that need to be addressed as part of the inclusion criteria. And then also, are you even engaging with first generation antihistamines? Are we focusing only on the second generation antihistamines? And finally, for inducible urticaria, it's very important to define what is going to be a washout before the provocation test. Usually, when it comes to second generation of antihistamines, we're talking about at least 3 days before the provocation test. They should not be on any antihistamine. And then from an exclusion criteria perspective, as you know, we have patients that overlap in terms of presence of both spontaneous and inducible urticaria. Do you really want these patients in your study if you're focused on one of the two? We're talking about 10 to 30% depending on which article you're reading. And then you have to be very careful about historical presence of other urticaria-looking skin diseases. The other area of debate is what do we do with IgE, and does it really make sense for all mechanisms of actions, right? Or when we're talking about those for targeting IgE. So do we want to define the screening levels? Do we want to cap patients who have screening levels of IgE below 100? Do we want to say only patients? We've had actually such study where the sponsor was focused on patients who have only IgE above 100. The problem is that your screen failure rate goes through the roof. So something to also keep in mind. From an operational perspective, working with PROs has its advantages, but has significant potential challenges as well. So preparing the patient and educating the patients how to properly do this, and then keep patients engaged throughout the study to make sure that you have the data that you need in order to make informed and accurate decision is not always easy. And there are 2 options. You can go with the paper or you can go electronic, and you pick the last 2 evils, right? So from a paper perspective, clearly it's more simple, it is easily accessible, definitely lower cost, so maybe for your small studies, very few sites have great engagement, they know the patients, this can work, but there is obviously important risk of data entry errors, not entering data. We all know patients who are sitting or just about to enter the investigator's office and they're filling out the paper on the right. So this is extremely dangerous for this particular indication because this is our bread and butter. This is what's going to decide the future of your asset. So reduced compliance is really something that unfortunately we've seen, and there is administrative burden, right? On the other side, electronic PROs look very nice when you listen to different vendors, right? When you start working with them, there are some challenges as well. So definitely we'll have improved data quality, increased compliance because they get reminded. It's almost real time, right? Prevent possible backdating, which is the problem with paper, and the regulators do prefer it. But on the other side, there can be glitches, right? From a provocation test perspective, and now I'm speaking again not only from the literature but from the personal experience, the most important part is actually training the sites. It is not complex, but the sites are not used to it, especially if you want to use temp test and freak test. So the best way to do it is to, and you should, if you're an investigator, because I see quite a few investigators in the room and sponsors who know this, is you really want during investigator meetings to have hands-on training, where the investigators are going to try on themselves and their staff is going to try on themselves to see how does it feel, what is the effect, and know how to actually consistently, accurately, reliably implement it during the study. What are the locations of the test? The locations can be different depending on whether it's a cold urticaria, it's primarily going to be lower part of your arms, but for example, for cholinergic urticaria demographies, you can do it on your back as well. So training material is the key, and we've learned this through our studies, is really what are also the weaknesses. So it's important to incorporate all the potential pitfalls in order to get the high quality of the data. The next big challenge is becoming patient recruitment. I was actually talking to chief medical officers this morning and they told me the same thing, right? That patients are becoming scarce. So it's not a little topic, and especially sometimes the screen failure rate in these studies can be high. So even when you have an initial impression that you'll be fine, by the time all patients are screened, you see that the challenge continues. So the site databases have to be capitalized on, for sure, but in addition to that, this is a physician referral indication. Reason, these patients are often treated by general practitioners, so connecting with your local hospital, connecting with your local multi-group office would help you in engaging these patients. In addition to that, obviously, when you're working with a CRO, there will be a proper central and local advertising plan. Sometimes central makes sense, sometimes doesn't make sense, depending on the size of the studies. Actually, Martin mentioned one of the registries. You should capitalize on registries in chronic urticaria as well as patient advocacy organizations. And by the way, they're mushrooming now. I was just Googling last night to see it, just to make sure that I'm up to date on everything. And I discovered another patient advocacy group called BQU, and it was funded late last year. So I have to look them up seriously to see what they're about. But what I'm trying to tell you is you need to be up to date with this. And then related to patient accessible information, you've noticed from both of our previous speakers that we're still not diagnosing necessarily chronic urticaria on time. So there is a need to raise awareness around what urticaria is. And actually there were some articles published about how can we target females in particular, because there is this 2-to-1 ratio when it comes to chronic urticaria. So developing some sentencing materials could make a difference in terms of patient recruitment. And then the next big one is site selection, right? When you're a site, you want to be selected. When you are a sponsor, you want to select the best sites. I tend to be one of those who believe that although chronic urticaria is easier to diagnose, we prefer to recommend the dermatologists and allergists to be investigators in the studies. However, I will say that there are practitioners of other specialties who've been in chronic urticaria space for years. I think we should feel comfortable with those, but outside of that, I would be careful, right, in terms of the choice. But when it comes to site selection, what sites are going to be interested in is, how is this study going to impact the patient's interest in the actual protocol and how it's going to impact their work. So we really need to optimize our studies. We need to pay attention to what is the prevalence of a particular type of urticaria in a certain region. As you probably know, environmental factors do matter. We've noticed within our studies, but it's in the literature as well, that in colder areas you will have more patients with cold urticaria. bigger. Yeah. So, and then experience size is what I'm trying to say, and in terms of really collaboration with the sites, I think it's more than ever important to collaborate closely with coordinators, because coordinators stay in touch with patients. Patients need to be extremely engaged in these studies because they're giving us all the data that will allow us to have a positive study. So what we all in this room are doing, we're making an impact in chronic urticaria space, and it's tremendously important, and I really have the highest respect for all of you in the room for doing that. And I would like us all to aspire towards this. Disease-modifying treatment criteria, and you developing disease-modifying treatments. The reason why this is going to be important is when you look at the long list of treatments that are in development, you need to differentiate. You need to have a differentiating target product profile in order to one day be able to precisely treat certain patients. I love this article and I wholeheartedly recommend it. Marcus was the first author and Martin is the senior author of this article, and it's explaining to us how should we define what is disease modifying in chronic urticaria. So from the point that it's preventing progression of the disease, so it's delaying it or it's slowing it down, to how we define remission. Is it 1 year? Is it 5 years? Is it without treatment? Is it with only antihistamines? For how long? Is it short term? Is it long term? And then can we dare to hope that one day we will have the cure? And how do we define the cure? And I really want to leave you with that. And hopefully this is what we are all striving for, to design and bring to patients disease-modifying solutions for chronic urticaria. Thank you.